Hello students, today we'll study about protoplast culture. This is the second video of protoplast culture. So let's begin. Somatic protoplast culture and somatic hybridization. So today we'll learn in this video about totally wholly somatic hybridization and protoplast culture. So somatic hybridization involves three aspects. First is fusion of protoplast. Second is selection of hybrid cells. Third is identification of hybrid plants. Now, first is fusion of plants. Fusion of protoplast. So, as the isolated protoplast are devoid of cell walls, in their in vitro fusion becomes relatively easy. There are no barriers of incompatibility for the protoplast fusion. Protoplast fusion involves mixing of protoplast of two different genomes can be achieved by spontaneous mechanical or induced fusion method. Now let's know about spontaneous fusion. Cell fusion in a natural process as is observed in case of egg fertilization. During the course of enzymatic degradation of cell walls, some of the adjoining protoplast may fuse to form homokaryocytes or homokaryons. These fused cells may sometimes contain high number of nuclei, that means 2 to 40. This is mainly because of expansion and subsequent coalescence of plasmodermal connections between cells. The frequency of homokaryon formation was found to be high in protoplast isolated from dividing cultured cells. Spontaneously fused protoplast, however, cannot regenerate into whole plants except undergoing a few cell divisions. Let's know about mechanical fusion. The protoplast can be pushed together mechanically to fuse protoplast of Lilium and Trillium in enzyme solutions can be fused by gentle trapping in a depression slide. Mechanical fusion may damage protoplast by causing injuries. Now let's know about induced fusion. Freshly isolated protoplast can be fused by induction. There are several fusion inducing agents which are collectively referred to as fusogens. Example NaNO3, high pH per calcium Ca2+, polyethylene glycol, polyvinyl alcohol, lysozyme, concavalin A, dextran sulfate, fatty acids, esters, electrofusion. Some of the fusogens and their use in induced fusion are now we will discuss. Now about sodium nitrate treatment. The isolated protoplast are exposed to a mixture of 5.5% NaNO3 in 10% sucrose solution. Incubation is carried out for 5 minutes at 35 degrees centigrade followed by centrifugation. That should be 200 Xg for 5 minutes. The protoplast pellet is kept in a water bath at 30 degrees centigrade for about 30 minutes, during which period protoplast fusion occurs. NaNO3 treatment results in a low frequency of heterokaryon formation, particularly when mesophyll protoplasts are fused. Now let's know about the high pH and high calcium ion treatment. This method was first used for the fusion of tobacco protoplast and is now used for other plants too. The method consists of incubation protoplast in a solution of 0.4 m mannitol containing 0.05 m calcium chloride at pH 10.5. Glycine NaOH buffer and temperature 37 degrees centigrade for 30 to 40 minutes. The protoplast from aggregates and fusion usually occurs within 10 minutes. By this method, 20 to 50% of protoplasts are involved in fusion. Let's know about PEG treatment, polyethylene glycol treatment. This has become the method of choice due to its high success rate for the fusion of protoplast from many plant species. The isolated protoplast is in culture medium are mixed with equal volume in the tube. That means 1 ml of 28 to 56% PEG in the tube. PEG enhances fusion of protoplast in several species. This tube is second and then allowed to settle. The settled protoplasts are washed several times with culture medium. PEG treatment method is widely used for protoplast fusion as it has several advantages. Let's know about the advantages. First point is it results in a reproducible high frequency of heterokaryon formation. Second is low toxicity to cells. Third is reduced formation of binucleate heterokaryons. PEG-induced fusion is non-specific and therefore can be used for a wide range of plants. Now let's know about the electrofusion. What is the term electrofusion means? In electrofusion, this is a method in which electrical field is used for protoplast fusion. When the protoplasts are placed in a culture vessel fitted with the microelectrodes and an electrical shock is applied, protoplasts are induced to fusion. Electrofusion technique is simple, quick and efficient and hence preferred by many workers. Further, the cells formed due to electrofusion do not show cytotoxicity responses as is the case with the use of fusogens including PEG, polyethylene glycol. The major limitation of this method is the requirement of specialized and costly equipments. Now let's know about the second method, mechanism of fusion. What are the mechanism of fusion? The fusion of protoplast involves three phases: agglutination, plasma membrane fusion, and formation of heterokaryons. First, we should know about agglutination, or otherwise it's called as adhesion. When two protoplasts are in close contact with each other, adhesion occurs. Agglutination can be induced by fusogens. Example PEG, high pH and high calcium. Now let's know about the plasma membrane fusion. Protoplast membranes get fused at localized sites at the point of adhesion. This leads to the formation of cytoplasmic bridges between protoplasts. The plasma membrane fusion can be increased by high pH and high calcium, high temperature and PEG. Let's know about the details. That means high pH and high calcium ions neutralize the surface charges on the protoplast. This allows closer contact and membrane fusion between agglutinated protoplast. High temperature helps in the intermingling of lipid molecules of agglutinated protoplast membranes so that membrane fusion occurs. PEG causes rapid agglutination and formation of clumps of protoplast. This results in the formation of tight adhesions of membranes and consequently their fusion. Now let's know about the formation of heterokaryons. The fused protoplast get reduced as a result of cytoplasmic bridges leading to the formation of spherical homokaryons or heterokaryons. Let's know about selection of hybrid cells. About 20 to 25% of the protoplast are actually involved in the fusion. After the fusion process, the protoplast population consists of a heterogeneous mixture of unfused protoplast, unfused chloroplast, homokaryons and heterokaryons. It is therefore necessary to select the hybrid cells or heterokaryons. The commonly used methods employed for the selection of hybrid cells are biochemical, visual and cytometric methods. Now let's know about biochemical methods. The biochemical methods for selection of hybrid cells are based on the use of biochemical compounds in the medium. These compounds help to sort out the hybrid and parental cells based on the differences in the expression of characters. Drug sensitivity and auto and auxotrophic mutant selection methods. Now we will discuss. First is drug sensitivity. This method is useful for the selection of hybrids of two plant species. If one of them is sensitive to a drug, protoplast of Petunia hybrid, that is, let's say species A, can form microscopic callus on MS medium, but are sensitive to actinomycin D. Petunia parodi protoplast, let's say species B, form small colonies but are resistant to actinomycin D. When these two species are fused, the fused protoplast derive both the characters. The formation of microscopic colonies and resistant to actinomycin D on MS medium. This helps in the selection of hybrids. The parental protoplast of both species fail to grow. Protoplast of P. parodi form very small colonies, while that of P. hybrida are inhibited by actinomycin D. Drug sensitivity technique was originally developed by the scientist named Power et al. In 1976 for the selection of hybrids of Petunia species. A similar procedure is in use for the selection of other somatic hybrids. Example, hybrids between Nicotiana sylvestris and Nicotiana nightiana. Now let's know about the second method, that is auxotrophic mutants. Auxotrophs are mutants that cannot grow on a minimal medium and therefore require specific compounds to be added to the medium. Nitrate reductase deficient mutants of tobacco are known. The parental protoplast of such species cannot grow with nitrate as the sole source of nitrogen, while the hybrids can grow. Two species of nitrate reductase deficiency, one due to lack of apoenzyme and the other due to lack of molybdenum cofactor. The parental protoplast cannot grow on nitrate medium, while the hybrid protoplast can grow. The selection of autotrophic mutants is possible only if the hybrid cells can grow on a minimal medium. Another limitation of the technique is the paucity of higher plant auxotrophs. Now let's know about visual methods. Visual selection of hybrid cells, although tedious is very efficient. In some of the somatic hybridization experiments, chloroplast deficient protoplast of one parent are fused with green protoplast of another parent. This facilitates the visual identification of heterokaryons under light microscope. The heterokaryons are bigger and green in color, while the parental protoplast are either small or colorless. Further identification of these heterokaryons has to be carried out to develop the specific hybrid plants. There are two approaches in this direction: growth on selection medium and mechanical isolation. Let's know about the visual selection coupled with Differential media growth. There exist certain natural differences in the sensitivity of protoplast to the nutrients of a given medium. Thus, some media can selectively support the development of hybrids but not the parental protoplast. Now let's know about the mechanical isolation. The visually identified heterokaryons under the microscope can be isolated by mechanical means. This involves the use of the special pipette, namely Drummond pipette. The so isolated heterokaryons can be cloned to finally produce somatic hybrid plants. The major limitations of this method is that each type of hybrid can require a special culture medium for its growth. This can be overcome by employing microdrop culture of single cells using feeder layers. Now let's know about the cytometric methods. Some workers use flow cytometry and fluorescence activated cell sorting techniques for the analysis of plant protoplast. While their visibility is maintained, the same techniques can also be applied for sorting and selection of heterokaryons. The hybrid cells derived from such selections have proved useful for the development of certain somatic hybrid plants. Let's know about the identification of hybrid plants. How do you identify the hybrid plants? The development of hybrid cells followed by the generation of hybrid plants requires a clear proof of genetic contribution from both the parental protoplast. The hybridity must be established only from euploid and not from uneuploid hybrids. Some of the commonly used approaches for the identification of hybrid plants are briefly now dis- we have discussed. Now let's know about the morphology of hybrid plants. Morphologically, the hybrid plants, which usually are intermediate between two parents, can be identified. For this purpose, the vegetative and the floral characters are considered. These include leaf shape, leaf area, root morphology, flower shape, its structure, size and color, and seed capsule morphology. The somatic hybrids such as pomatos and topatos, which are the fused products of potato and tomato. So, abnormal morphology and thus can be identified. Although the genetic basis, Although the genetic basis of morphological characters has not been clearly known, intermediate morphological features suggest that the traits are under the control of multiple genes. It is preferably to support hybrid morphological characters with evidence of genetic data. Now let's know about the isoenzyme analysis of hybrid plants. The multiple forms of an enzyme catalyzing the same reaction are referred to as isoenzymes. Electrophoretic patterns of isoenzymes have been widely used to verify hybridity. Somatic hybrids possess specific isoenzymes of one or the other parent or both the parents simultaneously. There are many enzymes possessing unique isoenzymes that can be used for the identification of somatic hybrids. Example, amylase, esterase, aspartate, aminotransferase, phosphodiesterase, isoperoxidase, and hydrogenase of alcohol, lactate, and malate. If the enzyme is dimeric, that means having two subunits, somatic hybrids usually contain an isoenzyme with an intermediate mobility properties. The isoenzymes are often variable within the same plant. Therefore, it is necessary to use the same enzyme from each plant. That means parents and somatic hybrids from a specific tissue with the same age. Let's know about the chromosomal constitution. The number of chromosomes present in the hybrid cells can be directly counted. This provides information on the ploidy or the ploidy state of the cells. The somatic hybrids are expected to possess chromosomes that are equal to the total number of chromosomes originally present in the parental protoplast. Sometimes the hybrids are found to contain more chromosomes than the total of both the parents. The presence of chromosomal markers is greatly useful for the genetic analysis of hybrid cells. Now let's know about molecular techniques. Many recent developments of molecular biology have improved the understanding of genetic constitution of somatic plant hybrids. Some of them we will discuss. Differences in the first point is differences in the restriction patterns of chloroplast and mitochondrial DNAs. Second point is molecular markers such as RFLP, AFLP, RAPD and microsatellites. Third is PCR techniques. These are the three techniques. These are the molecular techniques from which we can know the genetic constitution of somatic plant hybrids. Now let's know about the chromosome number in somatic hybrids. The chromosome number in somatic hybrids is generally more than the total number of both of the parental protoplast. However, wide variations are reported. Let's know about the reasons why. So, first is fusion of more than two protoplasts. Second is regularities in mitotic cell divisions. Third is infusion or electro-induced fusions. About 1/3 of the fusions occur between more than two protoplasts. Differences in the status of protoplast from the two species of plants result in formation of asymmetric hybrids. Next point is asymmetric hybrids may be due to unequal replication of DNA in the fusing protoplast. Protoplast isolation and culture may also lead to somaclonal variations, thus variations in chromosome number. Now let's know about the symmetric and asymmetric hybrids. If the chromosome number in the hybrid is the sum of the chromosome of the two parental protoplasts, the hybrid is said to be symmetric. Symmetric hybrids between incompatible species are usually sterile. This may be due to production of 3N hybrids by fusing 2N of one species with N of another species. Asymmetric hybrids have abnormal or wide variations in the chromosome number than the exact total of two species. These hybrids are usually formatted with full somatic complement of one parental species, while all or nearly all of the chromosomes of other parental species are lost during meiotic division. Asymmetric hybrids may be regarded as hybrid but for the introgressed genes. Protoplast fusion between N tabacum 2N equals to 48 and N nesophila 2N equals to 24 results in asymmetric hybrids, while asymmetric hybrids are formed when B napus and B zunia are fused. Now let's know about the hybrids. What do you mean by hybrids? The cytoplasmic hybrids, or in short hybrids, where the nucleus is derived from only one parent and the cytoplasm is derived from both parents, are referred to as the hybrids. The phenomenon of formation of hybrids is regarded as hybridization. Normally, hybrids are produced when protoplasts from two phytogenetically distinct species are fused. Genetically, hybrids are hybrids only for cytoplasmic traits. Hybrids and somatic incompatibility. Many a time, production of full-fledged hybrids through fusion of protoplast of distantly related higher plant species is rather difficult due to instability of dissimilar genomes in a common cytoplasm. This phenomenon is referred to as somatic incompatibility. Hybrids formed despite somatic incompatibility may exhibit structural and developmental abnormalities. Several generations may be required to eliminate the undesirable genes. Due to this limitation in hybridization involving protoplast fusion for partial genome transfer is gaining importance in recent years. Now let's know about the methodology of hybridization. How can we do hybridization? Okay, what is the procedure? Heterogaryons occur during hybridization. The nuclei can be stimulated to segregate so that one protoplast contribute nucleus alone or both nucleus and cytoplasm. In this way, hybridization can be achieved. Some of let's know about some approaches of hybridization. First approach is the protoplast of cytoplasm donor species are irradiated with X-ray or gamma rays. This treatment renders the protoplast inactivate and non-dividing, but they are efficient donors of cytoplasmic constituents when fused with recipient protoplast. Next point is normal protoplast can be directly fused with enucleated protoplast. Enucleated protoplast can be isolated by high-speed centrifugation. Next point is protoplasts are inactivated by metabolic inhibitors such as iodoacetate. In practice, iodoacetate treated protoplasts are fused with X-ray irradiated protoplast for more efficient formation of hybrids. It is possible to suppress nuclear division in some protoplast and fuse them with normal protoplast. Now let's know about the genetic recombination in asexual or sterile plants. There are many plants that cannot reproduce sexually. Somatic hybridization is a novel approach through which two parental genomes of a sexual or sterile plants can be brought together. Thus, by fusing parental protoplast, fertile diploids and polyploids can be produced. Now let's know about the overcoming barriers of sexual incompatibilities. Sexual crossing between two different species and two different genus is impossible by conventional breeding methods. Somatic hybridization overcomes the sexual incompatibility barriers. Let's know brief in two examples. First example, fusion between protoplast of potato, Solanum tuberosum, and tomato, Lycoperdicon esculatum, has called pomato, Solanopericon, a new genus. Next example, this is one example. Next second example, interspecific fusion of four different species of rice, Oryza brassaintha, Oryza Ichnegeri, Oryza officinalis, and Oryza periri could be done to improve the crop. Now, let's know about the applications of hybrids. Hybridization is a wonderful technique wherein the desired cytoplasm can be transferred in a single step. Hybrids are important for the transfer of cytoplasmic male sterility, antibiotic and herbicide resistant in agriculturally useful plants. Some of the genetic traits in certain plants are cytoplasmically controlled. This includes some types of male sterility resistant to certain antibiotics and herbicides. Hybridization has been successfully used to transfer CMS in rice. Hybrids of Brassica raffinus that contain nucleus of B. napus, chloroplast of atrazine resistant B. campus, resistant B. campestris, and male sterility from Raffinus sativas have been developed. Now let's know about the application of somatic hybridization. Somatic hybridization has opened new possibilities for the in vitro genetic manipulation of plants to improve the crops. Somatic hybridization has opened new possibilities for the in vitro genetic manipulation of plants to improve the crops. First is disease resistant. The plant will be disease resistant. Several interspecific and intergeneric hybrids with disease resistant have been created. Many disease resistant genes, example tobacco mosaic virus, potato virus, clove rot disease, etc., could be successfully transferred from one species to another. For example, resistance has been introduced in tomato against diseases such as TMB, that means tobacco mosaic virus, spotted wilt virus, and insect pest. Now let's know next disease is next point is environmental tolerance. The genes responsible for the tolerance of cold, frost, and salt could be successfully introduced through somatic hybridization. Example: introduction of cold tolerance gene in tomato. Next point is quality characters. Somatic hybrids for the production of high nicotine content and low erucic acid have been developed. Now, next point is cytoplasmic male sterility. A modification of hybridization in the form of hybridization has made it possible to transfer cytoplasmic male sterility. Now, let's know other applications of somatic hybridization. Somatic hybridization has helped to study the cytoplasmic genes and their functions. In fact, the formation is successfully used in plant breeding programs. Protoplast. Next point is protoplast fusion will help in the combination of mitochondria and chloroplast to result in a unique clear cytoplasmic genetic combination. Next point is somatic hybridization can be done in plants that are still in juvenile phase. Next point is protoplast transformation followed by somatic hybridization will yield innovative plants. Now let's know about the limitations of somatic hybridization. Although somatic hybridization is a novel approach in plant biotechnology, there are several problems and limitations. The success of the technique largely depends on overcoming some limitations. Let's know about the limitation. First point is somatic hybridization does not always produce plants that give fertile and visible seeds. Next point. Second point is regenerated plants obtained from somatic hybridization are often variable due to somaclonal variation, chromosomal elimination, organelle segregation. Next point is protoplast culture is frequently associated with genetic instability. Next fourth point is protoplast fusion between different species or genus is easy, but the production of viable somatic hybrids is not possible in all instances. Next point is some of the somatic hybrids, particularly when produced by the fusion of taxonomically different patterns, are unbalanced and not viable. Next point is there are limitations in the selection methods of hybrids as many of them are not efficient. Next point is there is no certainty as regards the expression of any specific character in somatic hybridization. Next point is somatic hybridization between two diploids results in the formation of an ampidiploid which is not favorable. For this reason, haploid protoplasts are recommended in somatic hybridization. Finally completed. Any questions regarding this, regarding protoplast culture and somatic hybridization, you can ask me in the comment box and subscribe my channel. Thank you.